So now we are ready to analyze our kinetics data that we obtained during last week's lab. Okay, so a little bit of background, and then we're going to walk through how we are going to use the data that we obtained to actually determine some important kinetics parameters from our rate law. So remembering, when you have a generic chemical reaction or a reaction and you want to write a rate law, which you write is rate equals rate constant times the concentration of each reactant raised to powers that we call orders. And it's important to highlight that when writing a rate law, you only use reactants in writing a rate law. And importantly, these reaction orders must be determined experimentally. Remember, those reaction orders tell us how the rate will change when concentrations are changed. Is it a direct relationship when we have a reaction order of 1, or it is a square relationship or a cube relationship if x is equal to 2? Really, we don't have 3, or is it there no dependence, and that's a 0 order process? Okay. What we've done a lot in lecture is determine X and Y using a process called the method of initial rates. And what that means is we have multiple experiments where we're going to be holding one concentration constant while we're changing another. So for example, if we hold the concentration of A constant and then we change the concentration of B, we're going to see how the rate changes, and that's going to determine this reaction order with respect to B, which is going to be Y. We can do the reverse experiment where we're going to hold the concentration of B constant, change the concentration of A, and then we'll monitor how the rate changes, and that will allow us to determine the reaction order with respect to A, which is X. So that's what we're doing a lot of in lecture. We're actually going to do something different here in lab. There is another way that you can determine reaction orders. So this is a slide from my lecture slide deck that just kind of shows us when you plot reaction data, depending on how you plot it to give a straight line, that can be used to determine reaction order. So just take a look at this for a second. Okay, we're always going to be plotting something versus time, some way of measuring the quantity of our reactant as a function of time. And whether we plot the natural log, 1 over the concentration, or the concentration without any sort of manipulation, depending on which one of these gives a straight line, that's going to determine reaction order. So plotting the natural log versus time, if that gives a straight line, that tells us that that reaction is first order with respect to that species. Second order will have a straight line when you plot 1 over the reactant concentration as a function of time. And if you get a straight line when you just show the reactant concentration as a function of time, that's going to be 0 order. So we can use graphical data to determine reaction order. And that's what we're going to do in this lab. Okay. So how do we connect this to our lab? Remembering, because it may have been a week or two since you've done the data collection, this lab was using a blue dye and mixing it with the hypochlorite ion, which is the active ingredient in bleach, and bleaching that dye to get a colorless product. So we're measuring the blueness of that dye using a spectrophotometer. So Beer's law, remember Beer's law is what allows us to directly correlate the absorbance of a species with its concentration. What we can do is we can take that absolute number we get when we measure absorbance, and we can use that as a direct correlate for what the concentration of blue dye is. Okay, so we're going to be able to use the information we get directly from the spectrophotometer, which is measuring the absorbance at 630 nanometers, and we're going to essentially plot that as concentration of our blue dye, because Beer's law tells us that these are linearly correlated. Okay, so what we're going to do then, just like I showed you on the last slide, if blue dye, if that is one of our reactants, okay, we can plot the concentration of blue dye versus time, or the natural log of the blue concentration versus time, or 1 over the blue dye concentration versus time. And we're going to see which one of those gives us a straight line, and then we're going to use the slope to tell us the rate constant. So I know there's a lot to unpack there, so let's walk through it a little bit more slowly and talk about exactly what you're going to do with your data. Okay, when you downloaded your data from that little spectrophotometer, you had an Excel file that essentially had at least 4, maybe more than that if you had to repeat a trial. You had trials of data, which would give you time in seconds and absorbance data for that specific time. Remember, we collected data every second for up to 200 seconds. Okay. So you have this data that you're going to open up in Excel, and then you're going to create what we call a scatter plot, which is plotting Y versus X, not connecting the lines together. Okay. And when you take the data that's given directly from the spectrophotometer, which is absorbance and time, and you plot absorbance versus time, you're going to see what kind of line that gives you. Okay. We're going to learn how in Excel to label that graph. We're going to call it absorbance versus time. We're going to label the axes so we know that it's time in seconds. Absorbance values have no units, okay, but we're going to label this absorbance, but we're going to remember that it's really reflecting the concentration of that blue dye. Okay. Then what we're going to do is we're going to add a trend line. We're going to learn how to do that. A trend line is basically asking Microsoft Excel to connect all the dots with a linear line, or you can have different kinds of lines. We're going to choose a linear line and we're going to say, how well does a linear line fit our data? That's going to be reflected in this R squared value. And then we're also going to have it plot the equation for that line, because that's going to be important, because the equation of a line, y equals mx plus b, the slope is a piece of information that we're going to want, because the slope of the line is going to give us the rate constant. And don't worry if I'm losing you a little bit here. We're going to go through this a lot, and we're going to do it together in lab so that everybody will kind of get to the same point. Okay, so you're going to do that for your trial 1 data. Now, we also need to do a little bit of data manipulation here. You're going to take this same data and you're going to take, and we're going to show you how to do this in Excel, you're going to take this absorbance data and you're going to create a new column of data, which is the natural log of that absorbance value. Okay, we're going to learn how to do that in Excel, but you're going to create a new column of data, and then you're going to create a new plot, which is now the natural log of that absorbance data versus time. Again, you're going to label it natural log of absorbance versus time. You're going to label your axes. You're going to add your trend line with your equation and your R squared value. And we're going to ask, how well does that data fit a straight line? Moving a little bit more quickly here, hopefully you see the variation on the theme. The last thing we're going to do is we're going to take this absorbance data and we're going to manipulate it and make a new column of data that is 1 over that absorbance value. And then we're going to create a third scatter plot. Call that 1 over absorbance versus time. Okay, plotting 1 over absorbance as a function of time, adding in our axes labels, putting in our trend line and our equation and our R squared value. And we get 3 plots for trial 1. You had 4 trials. So you need to do this for all 4 trials. And yes, that means you are going to have 12 plots that you make in Excel. Okay, everyone is going to need to submit their own Excel workbook on Blackboard. Don't submit it yet, because there's still more that we need to do, still more data manipulation that we're going to do in Microsoft Excel. Okay, so you made these 12 plots. What do we do with those 12 plots? What kind of information are we going to get? How do I use this data? Well, let me step back for a second and remind us of the rate law for our specific experiment that we're doing. Rate equals rate constant times the concentration of our blue dye raised to a power that is its order, times the concentration of the bleach hypochlorite ion raised to a power that is its order. Okay, so here's what we did here. We knew we had a really high concentration of bleach compared to the concentration of dye. Essentially, so that changing this concentration of bleach was going to be sort of minimal. So we could sort of simplify this rate law to be rate equals rate constant times the concentration of the blue dye raised to its power, that is its order. And what we really did is we put this term and this term together in a new rate constant. So this rate constant is really equal to the overall rate constant times the concentration of the bleach raised to a power that reflects its order. Okay? Well, why is this important? This allows us to sort of disentangle these two pieces so that we can use some experimental data to solve for X and other experimental data to solve for Y. Okay, so how do I use those 12 graphs that I just made? Well, it has to do with solving for x and this rate constant. So let's talk about how you do that. Okay, remember we are going to have 3 plots of data for each trial. Okay, you're going to look at those plots of data, and based on that r squared value, the closer the number is to 1, the more that data is a linear fit. Okay, so you're going to look at which one of those 3 plots, just looking at the blue die, the natural log of the blue die, or 1 over the blue die, and say which one is the best linear fit. Okay, if it's absorbance versus time, that means it's 0 order, and x here is equal to 0. If it's the natural log of the absorbance versus time, then that's first order, and x is equal to 1. If it's 1 over the absorbance data, that tells us that it's second order, and x is equal to 2. So we're going to look at the shape of those curves, find the one that best fits a linear fit, and that's going to tell us the reaction order with respect to our blue die. That's going to tell us our x value. So that's sort of part 1 here. The second part is once you figure out which one is the straight line, then you're going to take the trend line, which has an equation, and that equation, y equals mx plus b, m is equal to the slope. So you're going to take that number, that's your slope, that's your k, and it's going to have units of reciprocal seconds. Think about what slope is. Slope is a change in y over a change in x. We have something that has no units as absorbance values over seconds. So our units for our slope are reciprocal seconds, which are also going to be the units of our rate constant. Okay? All right. So you're going to do this again for each of the 4 trials. Each of the 4 trials is going to have a plot that looks the straightest, that's going to give you the reaction order. Now, as a heads up, they should be the same one. You shouldn't have one trial that tells you that it's 0 order, and then another trial.
Is there something else I can help with?
Nope, Sarah, I don't need help right now.
I don't have an answer for that. Is there something else I can help with?
No. Sorry about that. So all 4 of your trials should be internally consistent. You shouldn't have one trial that tells you it's 0 order, another one that tells you it's first order, and another one that tells you it's second order. That's not likely, okay? But you do need to do this analysis of looking at the slope for all 4 trials, and you're going to get 4 rate constants, okay? 4 rate constants, and I can show you where this stuff is going to go. The last page here, okay, we're going to have this page here where it has for your 4 trials. You're going to write what your slope is, and that same number is going to be your rate constant because your slope is your rate constant. Let me just go back to this slide really quick here. This slide here. Oops, this one here. Remember, do you see for all of these, the slope of that straight line is going to tell us k. Now, maybe it's minus k, but that slope is going to tell us k. Okay, so coming back here now. All right. All right, so that's how we're going to get pieces of information for this. It's going to tell us x, and it's going to tell us this sort of merged together rate constant. Okay, so let me flip back here just for a second to remind us, okay, we're going to use this data that we just talked about to give us x and then this merged together rate constant. We're going to use another set of data and another graphical analysis with this guy to tell us what y is and then what our overall rate constant is. Okay, so here's how we're going to do that. Okay, so kind of summarizing this, this was our overall rate expression for the reaction that we did. Okay, because these concentrations were so different, we were able to sort of merge this together and have an effective reaction rate that was only going to be reflecting our concentration order, or the order with respect to our blue dye. And now we're going to kind of use another set of data to determine y, and then also our overall rate constant. Okay, so stick with me here. We're going to talk about how we do that. Okay, the first thing we're going to need to do though is, because we're going to be talking about hypochlorite ion concentration, we're actually going to need to calculate what that concentration is. So this is just sort of a repeat of the table that you had when you're putting your experiment together, remembering that we always had the same volume of blue dye, so the concentration of blue dye was the same. You're going to actually need to calculate what that is. I'll give you a little bit of a hint. These are just M1V1 calculations. I'm going to show you where you put this on your handout here. It's going to be page 5. There's a spot where you can put your calculations in. And then data table 2 has concentrations for your blue dye and then your hypochlorite ion. So noting that this number is going to be the same for all of them, because we always added 1 milliliter of dye and it ended up being diluted to a total volume of 3 milliliters. Now, you have to do separate calculations for your bleach solution for that hypochlorite ion, because in this case, sometimes it was 2 milliliters diluted to 3, but then it was 1.5 to 3, 1 to 3, 0.5 to 3. So we're going to have decreasing concentrations of our hypochlorite ions. So you need to go and calculate those, because this green data is going to be stuff that we're going to need to do our next analysis. Okay, so make sure that that goes in this table here, because we're going to need to create a new worksheet in our Excel workbook. So a workbook has multiple worksheets. We were using one worksheet to do our last. We're going to add a new worksheet and do the next set of data analyses. Okay, so create a new worksheet in Excel, and you're going to have to manually create this table of data. You're going to have 4 trials worth of data. You're going to put in those bleach ion concentrations, that hypochlorite ion. So all of these numbers that are here, okay, from table 2 are going to go right in there. And then those rate constants. So remember the stuff that we just did here. Back page, we took those slopes of the lines that were straight, and those gave us rate constants for each trial. Those go in here. Okay, now we're going to make some more plots. Okay, so now we're going to plot rate constant as a function of hypochlorite ion concentration. We want to make sure we label these appropriately. Title it rate constant versus hypochlorite ion concentration. We're always going to be doing scatter plots here. You always want to do scatter plots when you want to let Microsoft Excel make the line for you. Okay, so rate constants have units of reciprocal seconds. Then we've got our hypochlorite ion concentration, which is going to have units of molar. Okay, so you're going to plot this, and then you're going to also add a trend line and then get your equation and your R-squared value, and then ask the question, is this linear? If you plotted the rate constant as a function of the concentration of hypochlorite ion, and that is linear, that tells us that it's first order with respect to the hypochlorite ion. That's going to be y equal to 1. Okay, we need to check and see, well, maybe it's something else. So we need to make a new column of data. Watch how this works. We're going to take our hypochlorite concentrations and we're going to square them. We'll learn how to do this in Excel, but you're going to make these 4 values here that are essentially the square of the hypochlorite ion concentration. We're going to plot that data. Now it's going to be rate constant versus the square of the hypochlorite ion concentration. Label the plot rate constant versus hypochlorite ion concentration squared. Again, that's going to be that scatter plot, so we can add the trend line with the equation and the R-squared value. Ask the question, which one of these is linear? If this guy is linear, then we know that it's first order with respect to hypochlorite ion concentration. If this guy is linear, then it's second order with respect to hypochlorite ion concentration. Oh boy, I know your head's probably hurting now. This is a lot. The last little piece here, just like we did with our first stuff, once we found the situation that created a linear plot, the slope was important, okay? Whichever one of these is linear, use that one, and take the slope of that, and that's going to be our overall rate constant, okay? So that's going to be what kind of goes in this last part down here, okay? And remember, the units of that are going to be whatever your y and your x are, because slope is y over x. So it's either going to be, this is our y, which really has a seconds in the denominator, over x. So it's either going to be reciprocal seconds, reciprocal molar, or it's actually going to be reciprocal seconds with 2 molars that are reciprocal, 2 molars in the denominator. Okay, so make sure you have your right units for that overall rate constant. Okay, I know this was a lot. I've got one more slide here to sort of try to sum this up, okay? And hopefully as a way to review and summarize what we did. Okay, big picture for this experiment. We can write a rate law for a chemical process, not knowing anything about it if we write it generically. Rate equals rate constant times the concentration of each reactant raised to a power that is its order. Now, we did something unique with this experiment, is that we used graphing experimental data as a way to determine our reaction orders and then our overall rate constant. So what I've highlighted here, these are sort of the money pieces for a rate law. Those are the things that start out generically, but then you want to determine. Now, some other things that we used here is we used varying concentrations of our bleach to be able to do this experiment. So let's just go through one last time here and kind of summarize all this up so that hopefully this makes sense with what you did today. So we were able to break out our rate law into 2 separate parts to allow us to use one set of experimental data to solve for our reaction order with respect to our blue dye, and then a merged sort of rate constant that keeps the concentration of hypochlorite essentially equal. Okay? We plotted it 3 different ways with 4 trials of data. So that was your 12 different plots. And we said, which way of graphing this data gives us a straight line? And by answering that question, we will determine the order with respect to the blue dye. Okay? So by looking at the identity of the plot and saying which linear plot gave us the best linear fit, or I should say which plot gave us the best linear fit, that will determine our reaction order with respect to blue. But we also got important information from that because the slope of whatever plot was linear gave us this rate constant, okay? And that's important because we've got 4 rate constants for 4 trials that are paired up with 4 different concentrations of bleach. Okay, so that's why we have 4 rate constants. We did 4 trials from 4 different concentrations of bleach. So now we're going to move on and we're going to use this data, specifically these rate constants and these 4 different concentrations of bleach, to determine the reaction order with respect to that hypochlorite ion. Okay, so the second piece we sort of like teased out when we merged these rate constants, or we merged these constants together. We said that K, okay, this K is equal to merging these 2 pieces together. Okay, so we can kind of indicate that that's what those are, but by doing that, that allowed us to do a separate graphical analysis to independently determine what Y is, and then also our overall rate constant. Okay, so here's how we did that. We had to make our own data table because we were going to plot 4 different rate constants as a function of 4 different concentrations of hypochlorite or bleach ion. Okay, so this is where we get this data. We had to take the data from our first experiment, those different rate constants, and then we had to plot them versus the concentrations of hypochlorite. And we had 2 plots for that because we had to say sometimes let's look at plotting it as just a direct relationship between the hypochlorite ion concentration, or does it look better? Does it look more linear? Which way gives us a straight line? Do we plot it as the square of the hypochlorite ion concentration? Okay, so we needed to say which gives us a linear plot. And if the linear plot was from, we would determine what Y is. If we used it as a plot of just the direct concentration of hypochlorite ion, that was first order. If it was a function of the square relationship of the hypochlorite ion, then it was second order. Okay, and then you can take the slope from whichever plot gave you the best linear fit, and that is going to give you your overall rate constant. Okay, and then once we have all of these pieces, we know all of the parts of our rate law. Okay, and that's a specific rate law. We know the overall rate constant, we got that here. And we know X and Y because we use two different graphical analyses to determine that. Wow, I know that was a lot, but that's also why we're going to have this video up so that you can rewatch parts of it if you need to see it. But we're going to go through this data analysis all together so that hopefully it makes crystal clear sense. Enjoy.